This is Unit 1 of AP Biology, looking at cell components now, just individual cell parts and their functions today. The first one we're going to look at is the nucleus, which is the control center of the cell. It acts this way because it stores the cell's DNA, which is again how proteins are coded for and made, and proteins do many cell functions, and so in many ways this does operate as the control center. The nucleus is surrounded by a structure called the nuclear envelope. As you can see here, this purple envelope's double layered all the way around. Protects the inside of the nucleus from the cell, from the inside of the cell. The cytoplasm gives that DNA a buffer. It is composed of these little tunnels here. You can see a close-up of the tunnel here. These are called nuclear pores. These nuclear pores are there to allow the passage of materials in and out of them, materials like nutrients, RNA, as RNA is leaving the nucleus after it's been made. Obviously, the nucleus contains the DNA. A portion of the nucleus that is denser than other portions is called the nucleolus. The nucleolus is the place where RNA is usually made. It's also the place where ribosomes are constructed inside the nucleus. A little bit more about the DNA. DNA can be found in two phases. It can be found as chromatin or chromosomes. Chromatin is the looser form of the DNA. It is when the DNA looks kind of like this messed up plate of spaghetti. The cell is not, it's not that the DNA is not or that is not organized during this time. It's just the cell is not going to divide. This is the normal life of the cell. When the cell gets ready to divide, the DNA is condensed into these little structures called chromosomes. Only about 10% of the cell's life is spent in this phase, so the DNA condenses into chromosomes, makes it easier to deal with, to divide, getting two nice and equal sets when it comes to making two brand new cells. Another structure is the ribosome. These are cell particles that are made out of RNA and proteins. We won't go so far as to call them organelles, but they are found in all types of cells, prokaryotic, eukaryotic cells, and their function is to make proteins. They are the site of protein synthesis. All proteins are made on ribosomes. There are two types of ribosomes found in the cell. There are free ribosomes, and free ribosomes are free-floating in the cytoplasm. The proteins that they make are going to stay inside the cell. There are bound ribosomes, and these ribosomes are bound on the endoplasmic reticulum, which we'll get ready to talk about. These proteins are going to leave the cell. And so free ribosomes, the proteins stay inside. Bound ribosomes, the proteins are going to leave the cell. The way they do that is through this system called the endomembrane system. So here, these little red dots all over this endoplasmic reticulum here are the ribosomes that are bound. You can see that they've made some proteins here. This little purple dots are proteins. The vesicles, this little bubble, is going to detach and go to this structure called the Golgi body. And in the Golgi body, the proteins are tagged, and they are then released out of the cell via exocytosis. And so let's go into a little bit more detail on this. First, let's look at the endoplasmic reticulum, the ER. There are two types of ER. There's the smooth and rough ER. The smooth ER does a couple of things. It creates lipids for membranes like phospholipids and steroids. It also helps with carbohydrate breakdown like glycogen. Another function is to detoxify the blood. Um, Liver cells, for instance, have a lot of smooth endoplasmic reticulum. The rough endoplasmic reticulum, again, this is where ribosomes are the bound ribosomes are. So this is a structure that helps with protein synthesis, provides a safe place for the proteins to fold up. We talked about chaperonins in a previous video where that kind of thing took place. And so the protein then that is made on that ribosome is deposited inside the endoplasmic reticulum. The protein folds up and then leaves the endoplasmic reticulum via a structure called a vesicle, which is like a temporary storage bubble. Here's a picture of a vesicle here. All right. And then it goes to the Golgi apparatus, or the Golgi body, I called it earlier. Both are commonly used of this structure. The Golgi receives those transport vesicles from the ER, as you see here. It's receiving it. And at that point, it's going to take that protein that has been made and it's going to tag it. It's going to put a sugar molecule on it, make this what's called a glycoprotein at that point, sugar protein. And it is a way of marking the protein to where it's supposed to go once it leaves the cell. It's just like going to the post office and then putting a postage tag on it. It's going to such and such city and so forth. And at that point, the vesicle will form again and eject those proteins via exocytosis outside the cell, and then they'll enter into the bloodstream or whatever and go to wherever they're headed. Another structure that we need to look at is called the lysosome, sometimes considered part of this endomembrane system. The lysosome is kind of like a stomach for the cell. It digests particles that the cells intake in. It also recycles old molecules for use again. It's full of digestive enzymes. The inside of a lysosome has a different pH than the inside of the rest of the cell. And so in this case, you can see this little bit of food or whatever is being taken in via phagocytosis, and then the lysosome joins with that to digest that part of those food particles. And then the last organelle that we'll talk about in this video is the vacuole. Vacuoles are just temporary storage, or not temporary, more permanent storage areas inside the cell. Plants have a giant central vacuole that stores water, but animal cells can also have vacuoles to store any number of things, fat or water or sugar. Some organisms have vacuoles that have other purposes. These vacuoles are called contractile vacuoles. They actually act as little propulsion mechanisms where the water gets pulled in and then they push the water out to propel these little organisms through their environment.